Hi everybody, today we'll be discussing the electron transport chain for you. I have done an overview of energy production in the previous video. Please do watch it before this. Welcome to Dr. Ashiq's Conceptuals here. Our topic today is the electron transport chain. Everybody know that the site of electron transport chain is mitochondria. So let's discuss the structure of mitochondria first. You know that mitochondria is a double membrane structure and this is the outer membrane of mitochondria. And you can see the inner membrane of mitochondria, and what is important for inner membrane of mitochondria is that the electron transport chain, our today's discussion topic, is situated over the inner mitochondrial membrane. And what I am going to mark here in between the outer membrane and inner membrane here, can you see that space between that two membranes, we'll call that space as the intermembrane space. That space between the two membranes, outer membrane and inner membrane, is known as intermembrane space. So we can see a magnified image of mitochondria there, and we'll be seeing a small portion of the mitochondria. You can see the outer portion, the upward portion is the intermembrane space, and the inner portion is the mitochondrial matrix, the inner part of the matrix. And the membrane that is shown here is the inner mitochondrial membrane, the site of electron transport chain. And you can see so many special channels are present over the inner membrane there, and these channels all together we'll call it as electron transport system. And let's see the details of the electron transport system here. So you have to know why the electron transport system is present there and how it is working there. We shall discuss all together in very detail here. So before going to the details of that thing, I'm remembering you to subscribe my channel for the future videos there. So why that electron transport chain is present there? The electron transport chain is there for the production of ATPs there. And how it is working? I'll be drawing the electron transport chain for you here. What you are seeing is that inner mitochondrial membrane. I am drawing is the lipid bilayer membrane of that inner mitochondrial membrane. So and you can see the complex channels present here. You can see what I am drawing are that complex channels there. So let's discuss each channel first. You have to know that all these channels are enzyme complexes. They are enzymes, they are complex enzyme, big molecules of enzyme. This is the first, second, third, 4 and 5 complexes are there, and lipid soluble molecule is there, that is known as ubiquitin, is attached with the complex 2. And a special thing regarding that, there is in between the complex 3 and complex 4, there is a special molecule of cytochrome C is present there. The cytochrome C is a mobile electron transporter that is present between the complex 3 and complex 4. We'll discuss the details of the cytochrome C at that point in the electron transport chain. So you have to see what happened. You can see that here the NADH is coming to the complex 1, and this NADH is converted to NADH plus and 2 electrons. Here the NAD is losing H plus and electrons. From your chemistry knowledge, you know that what is the process of loss of H plus and electron? That is known as oxidation. The NADH is getting oxidized. What happened to this electron? You have to know that the complex 1 is electronegative. So it will attract that electron towards the complex 1. And what will happen to this electron? The ubiquitin molecule, it is more electronegative than the complex 1. So the ubiquitin is attracting that electron towards it. So that electron from the complex 1 will be reaching the ubiquitin. So you have to see that the ubiquitin is gaining electron. Ubiquitin is gaining electron. So what you call the process of gaining of electron, we can call it as a reduction process. So you can see here both oxidation and reduction is occurring. So the NADH is getting oxidized and the ubiquitin, it is getting reduced. So you can see a combination of oxidation and reduction, or it is a redox reaction that is happening there. So we can name that enzyme that involved here, that is a complex 1. How the name of the complex 1 will be coming? You can see it is known as NADH ubiquitin oxidoreductase. NADH is getting oxidized and ubiquitin is getting reduced. So that enzyme is known as NADH ubiquitin oxidoreductase. That's how the complex 1 is named. We'll be summarizing that thing before. So the complex 1, you know that everybody should know the complex 1 is known as NADH ubiquitin oxidoreductase, or simply we can call it as NADH dehydrogenase. So what happened to these electrons present in the ubiquitin? You have to know that the complex 3 is more electronegative than the ubiquitin. So that electrons will go to the complex 3. And from the complex 3, what will happen? You can see that there is a cytochrome is present there. The cytochrome, I already told you that cytochrome C is a mobile electron carrier that is situated between complex 3 and complex 4. So whenever the complex 3 is having an electron, cytochrome will move towards the complex 3 and accept the electron here. So the electrons will move towards the cytochrome C. So from the ubiquitin, it will go through the complex 3 and it will go to the cytochrome C. So what happens there? The ubiquitin is losing the electron and cytochrome C is the gaining the electron. So we can tell like ubiquitin is getting oxidized and cytochrome C is getting reduced. So what will be the name of the complex 3 enzyme? Same name. You can see that ubiquitin cytochrome C oxidoreductase, oxidation of ubiquitin and the reduction of cytochrome C is occurring there. So the complex 3 is known as ubiquitin cytochrome C oxidoreductase. So we shall add to our summary here that complex 3, what is the name of the complex 3 will be adding to the summary here. It is known as, what is the name? Ubiquitin cytochrome C oxidoreductase. We shall proceed further here. So you know that complex 4 will be more electronegative than the cytochrome C. So the electron from cytochrome C will be easily going into the complex 4. Here the most electronegative particle molecule in the electron transport chain is coming here. It is none other than your oxygen. The oxygen will be accepting the electron from complex 4 here. And what happens there? You know, everybody know this reaction. The oxygen will combine with hydrogen ions and electrons in the matrix to produce water there. So you know that electron from that complex 4 is going to that oxygen here. So you can see that here the cytochrome C is losing the electron and oxygen is accepting that thing. So it is oxidation for the cytochrome C and the reduction of the oxygen is occurring there. So the complex, it is known as cytochrome C oxidase. It is known as cytochrome C oxidase. We shall add to our summary here. The complex 4 is known as cytochrome C oxidase. So we have one another complex is left there, that is your complex 2 there. We have discussed already 3 complexes we have already discussed and complex 2 is left. And what happens, what is the speciality with the complex 2? The complex 2 receives the hydrogen from a different hydrogen donation there, that is from the FADH molecule. The FADH molecule will be converted to FAD plus, H plus and electrons. And since it is the loss of hydrogen and electron, we can call it as FADH is getting oxidized here with the help of complex 2. What happened? The electrons released from here, the electrons will be accepted by the complex 2 and then it will be traveled to the ubiquitin. And since the ubiquitin is getting the electron, it is getting reduced here. So what will be the name of complex 2? It will be known as FADH ubiquitin oxidoreductase. That is the specific name for that thing, FADH ubiquitin oxidoreductase. And you have to think about the FADH, from where or how the FADH is produced in our body. There is only one source there. You know that in the TCA cycle, there is a complex that is known as succinate is converted into fumarate, and that is a conversion in a TCA cycle there. And in this process, the FADH is produced. And this process in the TCA cycle is carried out by one complex that is known as succinate dehydrogenase. And since the succinate dehydrogenase producing FADH, the other name for the complex 2 is succinate dehydrogenase. We shall add that name to that summary figure here. The FADH is converted into FAD, H plus and electron, and that complex 2 is known as succinate dehydrogenase. These are the names of different 4 complexes, electron transport chain complexes are there. And we have to discuss something else that is present in the NCERT textbook here, that complex 1, complex 2 and complex 3 is having a special thing that is known as an iron sulfur compound. A metallic compound is there, that is iron sulfur compound is present in complex 1, 2 and 3. And instead of that thing, complex 1 needs flavin mononucleotide, that is FMN, as a cofactor here. And the complex 3 and complex 4 is having different cytochromes are there. Complex 3 is having cytochrome B and cytochrome C1, and complex 4 will be having cytochrome A and cytochrome A3. So these are the additional information you have to remember about these complexes here. And let's discuss how these complexes will be working and how this process of electron transport is occurring here in simpler format. So there are two different pathways are working there. So you have to see that in a simple line diagram here. I am drawing complex 1, complex 2, the ubiquitin, then there is a complex 3 and the complex 4. And the terminal electron acceptor here in this series is oxygen. I am adding that also there. So to the complex 1, who is coming? To the complex 1, the NADH will be donating the electron, that it will be going through 1 to ubiquitin, 3 and 4 and 2. You can see that 2 is not involved here. And when the FADH is donating that electrons, what will happen to that electron? This electron will go to the 2 first, then the ubiquitin, then 3, then 4, then to the oxygen. So 1 is not involved in case of FADH here. So that is a special thing about NADH and FADH. NADH pathway will be 1, 3, 4, then oxygen. FADH, the pathway will be 2, 3, 4, then the oxygen here. So you know that as the electronegativity will be increasing towards the end of that thing, and most electronegative thing will be oxygen and least electronegative thing will be complex 1 and complex 2. So you have to know that what will happen to that electron. This electron that will be reaching the first and second thing are high energy electrons. From when the electron is transmitting from the 1 to ubiquitin and the 2 to ubiquitin, what will happen to the energy of that electron? You have to know that the energy of the electron is released out. Energy of the electron is released out. Same way from the ubiquitin to 3 also there is energy release is occurring, and 3 to 4 also the energy release is occurring there. So all the energy of that electrons is released out as it is going through here, and we will be storing that energy here. We will be using that energy. And what is the purpose of this energy release here? We'll be discussing here. What happens is that here you have to know that this is the matrix, the inner part of the mitochondria, that is the outer part of the mitochondria, that is the intermembranous space there, and all this structure is present on the intermembranous space. And here there will be lot of H plus ions will be coming from that reduction of FADH and NADH, and this energy that is released by the electron transport is used to pump this H plus into the intermembranous space. This energy released from that electrons will be used to pump these hydrogen ions into the intermembranous space there. So we shall see how it will be occurring there. You have to know that there is a certain number of hydrogen ions. So the complex 1 will be pumping 4 hydrogen ions into the intermembranous space. Complex 3 will be pumping 4 hydrogen ions, and complex 4 will be pumping 2 hydrogen ions there. And you have to know, due to this pumping of that hydrogen ion, there will be high concentration of hydrogen ions in that intermembranous space and low concentration of hydrogen ions in the matrix will be there. And other thing is that complex 2 is not pumping any hydrogen ions there. Only 1, 3 and 4 is pumping here. So here what is important is that whenever an NADH is coming, NADH electron will go through complex 1, 3 and 4. You know that complex 1 will be pumping 4 hydrogen, complex 3, 4 and complex 4, 2. So total 10 hydrogen ions are pumped from that NADH here. As well as when the FADH will be coming, the electron will go through 2, 3 and 4. You know that 2 is not pumping any electrons, and it will be pumping only 2, 4 plus 2, that is only 6 hydrogen atoms only pumped by that FADH here. And according to our calculation, 4 hydrogen atoms are needed for production of 1 ATP. So that NADH is pumping 10 hydrogen atoms. So total 2.5 ATPs will be given by the NADH. And FADH will be having only 6 hydrogen atoms, that by 1.5 ATP here. There is a difference in NCERT textbook. NCERT is telling 3 ATP from NADH and 2 ATP from FADH there. But actual practice, NADH will be giving only 2.5 ATP and FADH will be giving 1.5 ATP here. Let's see the details of ATP production here. We already discussed this figure here. You know, this is a cross section of that mitochondria. You can see the mitochondrial matrix and intermembranous space, and this membrane is the inner mitochondrial membrane where all these channels, and these channels will be pumping the H plus ions into the intermembranous space. Due to the pumping, the H plus ion concentration in the intermembranous space is going on increasing. So there will be a very high concentration of H plus in the intermembranous space and low concentration of H plus in the matrix will be there. So we have to discuss a special structure here, that is your complex 5. We haven't discussed this thing yet, but it is the time for you to discuss this thing. And this complex 5 is otherwise known as ATP synthase enzyme complex, which is producing the ATP. We have to study this thing with an example, the example of a hydroelectric power plant. So I have discussed this example in your previous video. If you haven't watched that video, you have to watch it now. So it is a hydroelectric power plant for you. You know that how the hydroelectric power plant is working here. We will be having a reservoir which is filled with water, and the water will be kept in the reservoir under high pressure. So there will be a large amount of water will be there, and this water will be flowing through a penstock pipe, a large pipe, and it will be going into a turbine. What will happen to the turbine? Turbine will rotate, and this rotation of the turbine will generate energy in the generator, and that energy electricity is sent to all the above our countries there. So this is what is happening in a hydroelectric power plant. We can compare this thing to our system here. Instead of water, we are having high concentration of H plus ion in the intermembranous space of the mitochondria. And what will be doing with the high concentration of H plus? These H plus ions will be flowing through a channel. You can see there will be a channel will be there, and these H plus ions are flowing through that channel. So in the intermembranous space, we are having a high concentration of H plus ions, and in the matrix there is a low concentration of H plus ion. And here your complex 5 is there. You can see this is your complex 5, and this complex 5 will act as a channel there. You can see that is a channel. Complex 5 ATP synthase enzyme will act as a channel there, and H plus ion will come through that channel from the intermembranous space into that mitochondrial matrix there, like in a reservoir. You can see that here there will be high concentration of H plus ions in the intermembranous space will be there. These high H plus ions will be flowing through a channel there. So this is a channel there. I'm drawing, this is a channel there, and what will happen? So there will be a channel plus a turbine is there, and this will be your complex 5. So from the high concentration of H plus, the H plus ion will be flowing down, and it will hit the turbine, and the turbine will rotate, and this rotation of the turbine will be producing energy, same as in the hydroelectric power plant here. You have to know that the channel and the turbine together constituting your complex 5. So this is your complex 5 here. So the complex 5, I will be marking it, and you can see it is situated on the inner mitochondrial membrane there, and it will be having two parts there. That is the first part and second part. The first part is situated on the membrane, and the second part is protruding into that matrix there. So the protruding part, the membranous part, it is known as F0 particle, and matrix part it is known as F1 particle. The F0 particle will be acting as a channel. You can see the 0 hole in the center, so that will be acting as a channel, and F1 particle will be the turbine there. So what is happening there? You can see the same figure here. So here I will be drawing that there is a high concentration of H plus ion maintained in the intermembranous space, and this is a channel there, the channel otherwise known as F0 particle of complex 5, and that through that channel that H plus ions are flowing, and it will be hitting on the turbine there, that round thing, that is a turbine. So turbine is otherwise the F1 particle of complex 5. So when it will be hitting, it will rotate, and this rotation of the turbine will produce energy. It will produce energy. And what is the purpose of this energy? We can use the energy for our physical work there. So there will be an ADP molecule will be coming. So that is an adenosine base with a 2-phosphate molecule attached to it. And along with that, there is an inorganic phosphate. Pi is inorganic phosphate is coming. We will use the energy produced due to the rotation to make a bond between the phosphate and the inorganic phosphate of the ADP and inorganic phosphate, and we will convert the ADP and the inorganic phosphate into adenosine triphosphate ATP. That is our energy currency, and that's how we will be producing that ATP. And this third bond is a high energy bond there. And how will you create this energy? By the rotation of the paddle. And this is the very famous theory of ATP production, that is known as chemiosmotic hypothesis of energy production. This is known as chemiosmotic hypothesis of energy production. And here the high osmotic energy of that hydrogen ion concentration is converted into chemical energy of the ATP. It is converted into chemical energy of ATP. So let's wind up this session by summarizing this thing here. So in the summary, you have to see that all the channels here. So in the diagram, I have given all the channels, and we'll see that all these first four channels, 1, 2, 3, 4 and fourth channel, they constitute the electron transport chain, and the fifth channel is the ATP synthase complex here. We have studied this thing. And in the ETS, what is happening? The NADH and FADH will be coming. Both the NADH and FADH will be getting oxidized, and the removed electron is passed through various channels, and that will be giving energy for ADP to add the phosphate here. The addition of phosphate is known as phosphorylation here. So we are having two processes, one is oxidation, another one is phosphorylation. In this system, both the oxidation and phosphorylation is occurring together. So we will call this process as coupled oxidative phosphorylation, or simply we can call it as oxidative phosphorylation, because oxidation and phosphorylation is occurring together here. What you have to discuss next is the figure from your NCERT textbook. You may be very familiar with this figure. So this is the figure which is given in old NCERT textbook. But there is a problem with the figure is that there is many mistakes there. So that NCERT withdrawn this figure and NCERT replaced with another figure here. Let's discuss this figure. This figure is marking that intermembranous space, inner mitochondrial membrane and matrix there. So you have to take your textbook and see this thing. You can see the NADH and FADH. NADH is coming there. NADH is connected to that complex 1. It is giving that electron there. That electron is taken up by the second complex, that you can see that ubiquitin is taking that thing, and ubiquitin is transferring that electron into the complex 3 there. You can see that is arranged like 1 3 is arranged, and from the ubiquitin you can see cytochrome C there. Cytochrome C is taking that electron to the complex 4. From the complex 4 you can see that the oxygen is coming, and oxygen is taking up that electrons there. And the second thing, and you can see here the succinate is converted into fumarate. So that is producing the FADH, and this FADH is taken up by that complex 2, then to ubiquitin. Through the ubiquitin is reaching the complex 3. Through the cytochrome C it will be going to that complex 4 there, and then also it will be also joining with the oxygen there. The terminal electron acceptor is oxygen there. And finally NCERT is showing your complex 5 there, the F0 F1 particle or the ATP synthase molecule there. So that's about your electron transport chain. Hope you like the video, hope you understand this thing. Thank you for watching me. I am Dr. Ashiq with you. And if you have any doubts, any more queries regarding this topic, you have to comment below in that comment box there, whatever the thing. Don't forget to subscribe, and I'll be telling you one thing, that I'll be doing a summary video for this thing, which will be having all the high yield MCQs for you. And please do watch the summary videos, which will be doing in a 2-minute video. Thank you.